now move on to the most awaited event on today's agenda. It is the keynote address by the keynote speaker, Vidya Jyothi, Professor Prasad Katulanda. Let me now take the privilege to introduce Professor Katulanda. Vidya Jyothi, Professor Prasad Katulanda, is a professor in medicine, head of the Department of Clinical Medicine, University of Colombo. He is a consultant endocrinologist and diabetologist. Professor Katulanda obtained his MBBS from the University of Colombo and his PhD from the University of Oxford, United Kingdom. Professor Katulanda is the Honorary Vice President of the Endocrine Society of Sri Lanka and the Honorary Assistant Secretary to the Sri Lankan Medical Association and the Country Director for the Asian Collaboration for Excellence in Non-Communicable Disease Program. He is well known as an excellent researcher in non-communicable disease researcher, and his work has primarily been focused on treatments and mitigations of non-communicable diseases in Sri Lanka. And he has published a number of research papers with over 6,000 citations in the fields of endocrinology and diabetology. Professor Katulanda joined the NIFS, then IFS, in 1988 as a pre-university research associate, where he initiated his research career. Today, we are honored to have him with us on this special occasion as we celebrate the 40th anniversary of the NIFS, and he will be delivering a lecture on application of diabetes and non-communicable disease research of Sri Lankan populations. Over to you, sir.
Thank you very much. Good morning, everyone. First of all, let me thank the organizing committee for inviting me to speak to you all today. Honorable Dr. Sita Arambepola, State Minister of the Ministry of Skill Development, Vocational Education, Research and Innovation, the Secretary, Chairman NIFS Professor Atula Sumatipala, Director NIFS Senior Professor Ranjit Premalal de Silva, Co-Chair of the Organizing Committee Professor Deepal Subasinghe, and the Organizing Committee, ladies and gentlemen. Let me share my slides with you. So again, it's a great pleasure, again, a privilege to be speaking as the keynote speaker at the 40th anniversary of the commencement of the NIFS, which was initially started as IFS. It's like coming home. And let me start my talk on application of diabetes and NCD research to Sri Lankan population. So if you look at the need and types of health research, we need to identify health problems which causes mortality and the suffering to people. So you need population research, which we call also epidemiological research, to identify, quantify problems. And also in that process, we might get some insights to the causation, like the risk factors, etiology, and so on. And then we need more investigations, particularly lab-based investigations, where fundamental and laboratory research becomes important to identify molecular level mechanisms in the causation of disease, as well as in developing treatments like drugs, medications, and also diagnostic tools for detection of disease. And then comes clinical research, where we want to further understand how a particular treatment works or how a diagnostic tool is used best and whether it is useful or useless, all this comes under clinical research. And then where we further evaluate the usefulness of either treatments, evaluations, or even preventive measures at the implementation level, the implementation research becomes important. So let me first show you a few things to make it more contextually appropriate. And you can see here the population pyramid in 1981. See, very few old people lived at that time, and mostly they were others for children or young adults. And things changed like that. By 2001, this became, the shape change. And more older people started to be living in the society. And now if you take today, this may be even more. So we are a rapidly aging society. Along with demographic transitions, the epidemiological transitions also started to happen, particularly with the increasing in the living conditions, where things like malaria, which were killing hundreds of thousands of people and even changing our civilizations from like Anuradhapura and downwards, started to go away. And we have now successfully eradicated endogenous malaria. We see only imported cases today. And you can see what is happening to the diabetes at the level of the global context. From 2000 to today, in about 20 years, it has increased about 300%. And this is going to be further increasing by a few more decades. And this is the World Diabetes Map. Initially, diabetes was considered a problem of the West and industrialized countries and the white Caucasians. But epidemiological research proved otherwise, particularly towards the last 2 decades. This epidemiological study showed that South Asians are a very vulnerable population, particularly in the type 2 diabetes. We know diabetes is where the blood glucose remains higher than normal in our circulation, and it can lead to acute metabolic complications like diabetes coma, ketoacidosis, and also more commonly, particularly in the type 2 diabetes, long-standing high blood glucose leads to chronic organ dysfunction and subsequently failure. Things which are unique to diabetes, we know the diabetic retinopathy, which can cause even blindness, diabetic neuropathy, which causes loss of sensation of our feet and subsequently loss of feet, and even other issues like erectile dysfunction, which leads to more societal problems, and then the nephropathy, which leads to kidney failure. In addition, diabetes people are vulnerable to develop atherosclerotic disease, where their big blood vessels get clogged with cholesterol-rich deposits and then subsequent blockade, which can lead to heart attack or brain attack, we call a stroke. And also this can affect our blood vessels in the leg, where we can suddenly lose a limb. So it is, we call as multisystem disease. There are many types of diabetes. Type 1 is predominantly due to absolute insulin deficiency due to the damage occurring to our beta cells in the pancreas that produces insulin from antibodies that develops against these cells. So there's no insulin. Insulin is like a key which opens a lock, and this opening of the lock leads to the opening of door, and as a result the glucose enters cells. So when there's no key, the doors remain shut and the glucose remains in the circulation with high blood glucose. And this occurs predominantly in children and young adults due to an autoimmune process. The common type 2 diabetes, which predominantly affects old people, the problem is not the deficiency of insulin predominantly, it is the functioning of the insulin. So you have insulin, the key, you have the receptor or the lock, it doesn't fit properly initially. So there's insulin resistance, which leads to, again, non-opening of the gates and glucose being high in the circulation. Type 2 diabetes is predominantly occurring in adults associated with obesity, sedentary lifestyle, and due to insulin resistance coupled with insulin deficiency. And similar mechanisms leads to diabetes occurring in the pregnancy. We call it gestational diabetes with risk to the mother and the fetus. And there are other specific types of diabetes, some due to genetic defects of insulin production, some due to genetic defects of insulin action, some defects due to problems in the pancreas, which leads to the production of insulin, and some due to drugs and so on. These are rare ones. Coming to chronic non-communicable diseases is accounting to about 80% of deaths, particularly in the developing countries. And these 4 chronic NCDs, chronic respiratory disease, diabetes, cardiovascular disease, and lifestyle-related cancers are due to 4 preventable risk factors: tobacco smoking, unhealthy diet, lack of exercise, and excess consumption of alcohol. So I told you about this 80% business. And also, earlier NCDs occurred mostly in older people who were not contributing to the production of the country. But now we increasingly see NCDs and diabetes affecting the people who are younger, less than 70 years. And we call this premature deaths and premature suffering, and can affect the economy and the development of the country and the society. So when I embarked on my research career, this is before around 2004, actually we didn't have very robust countrywide data on the prevalence of diabetes. The World Health Organization and the International Diabetes Federation used data from urban slums in Thailand to calculate our diabetes prevalence in this famous paper. And so therefore our estimates and projections were not accurate. Therefore, there was a need for comprehensive epidemiological study. And also there was no nationally representative data on different types of diabetes. There was hardly any knowledge about genetics of diabetes in South Asians, including Sri Lankans. And these things were thought of as huge challenges due to the cost and lack of expertise. Also, we didn't know whether we were an obese society or not. And also, there was a lot of controversy about whether the way the WHO and the Western countries classified obesity is applicable to Sri Lankans and other South Asians. We conducted this large epidemiological study. Unfortunately, we could not do it in the North and the East at that time due to the existing war at that time. And you can see some pictures of our teams going into even rural areas, collecting data. And in this seminal paper in 2006, we published very important data for Sri Lanka and showed that about 1/10 of adult population has diabetes, and about 1/3 of them did not know diabetes. And this increased with age, which was a common knowledge, but showed very exponential increase with age in our population, and showed that there's still a marked difference between urban and rural areas. And we were able to map the diabetes prevalence in different provinces. And you can see that the prevalence nicely fits in with the level of urbanicity, with highest in the western, then central, southern, then Sabaragamuwa, Northwestern, North Central, and Uva. And also showed that in the interventions, we need to take account about the other distributions like increase in prevalence in the Sri Lankan Moor population. And also we needed to know more deeper to see the real picture, and looked at the pre-diabetes. And you can see the overall pre-diabetes, which accounts for all the people with abnormal fasting glucose in between the normal and the diabetes range, and what's called the impaired glucose tolerance, which is after giving glucose 75 grams as a drink, the 2-hour value, which is again intermediate. And all these together also accounted to about another 11%. So altogether, about 1/5 of the Sri Lankans were not having normal blood glucose regulation. Also, we did a lot of other statistics to find out risk factors. You can see the family history, in both men and women, the presence of family history makes you at risk more than 2 times. And also, you can see with the increase in body mass index, as shown in the x-axis, the prevalence increases exponentially, and particularly this jump of the prevalence occurs around a BMI of 18. BMI of 18 we might consider thin at modern context. And you can see the comparison with Western data. This is the famous nurses health study from United States. You see the real increase in these people happens around a BMI of around 25 compared to 18 in our people. So also we showed the importance of physical activity. You can see the most active group has the lowest prevalence compared to the active group, and the interaction of the physical activity and the body mass index. Here, this is the group with the lowest BMI and lowest waist circumference and highest level of physical activity. Compare that with the group with highest waist circumference and the highest lowest level of physical activity. See the difference in the diabetes prevalence. Coming to the genetics of diabetes, we know that the genotype is our, like the birth certificate, our genetic identity. And the phenotype is what manifests like our height, color, and the behavior, disease status, and so on. The genetically based diseases can occur due to chromosomal disorders, due to single gene disorders, or due to multiple gene disorders we call complex traits. Diabetes is a classic example. So why we should know about the diabetes or genes due to responsible for any other chronic NCD? If we know the risk, genetic risk, we can predict the disease early and even take correctable measures or preventive measures. And also we can better understand the pathophysiology at molecular level, and perhaps might also help in individualization of therapy as well as development of drug targets. We call it pharmacogenetics. There are 2 main methods of looking at diabetes genes. In the first method called candidate gene approach, we usually suspect that this gene, because its mechanism involves particularly a disease process. For instance, we might suspect that a gene that encodes for the insulin receptor might be responsible for diabetes because of its mechanism in the involvement of glucose control, and then look at that gene. We call this candidate gene approach. It is like you use one light to find a lost key. And by this way, it takes a long period to look for, find genes. Another novel technique that was coming up was called the genome-wide association studies, where you used like multiple lights to find a lost key. And here what we do is we rapidly scan for multiple genetic variations across sets of DNA samples from many people. And this is very useful for complex multiple gene polygenic disorders like diabetes, asthma, cancer, heart disease, hypertension. What is done is there are things called DNA chips, so microarrays, where hundreds of thousands of single nucleotides are embedded there. And when the sample is run, it detects abnormalities at a given time. And so we, using all these techniques, we embarked on our work on genetics of diabetes. And in addition to the study I have already showed, we also used other studies like the young diabetes study, where we recruited 1,000 sample of people who developed diabetes less than 40 years and above 16, and particularly because we thought that they might have higher risk genes. And then use many genetic methods like DNA extraction, PCR, real-time PCR. Everybody talks about these because of the COVID screening, and DNA sequencing, and the various chips like Illumina chip for Jiva studies. And then came up with some seminal papers like this paper on mitochondrial diabetes among South Asians and Sri Lankans, where we were able to show that about 1% of overall diabetes in the young is due to mitochondrial diabetes, and they have characteristic clinical features compared to the others. And also we did a lot of work on this maturity onset diabetes of the young in young onset diabetic patients. And also we used other methods to find out risk genes for type 2 diabetes, and also contributed to this very seminal papers which were published in esteemed journals like Nature Genetics on genome-wide scans, finding new loci for type 2 diabetes, which were also highlighted in the media at that time. And also we were able to contribute to knowledge where the different subtypes of diabetes in the Sri Lankan population. And you can see here different subtypes based on these autoantibodies and other molecular markers, type 2, and then various other subtypes, which I'm not going to details. Particularly when it comes to epidemiological research, just a one-off research won't be adequate because the epidemiology changes. You need to see the disease trends, patterns, changing risk factors. So in keeping with that need, we also repeated some of our work on epidemiology, and in the Sri Lanka non-communicable disease study, which we were unable to complete in the whole country due to the financial constraints. However, we managed to do in the Western province. You can see about 1/5 of the people already knew that they had got diabetes. And also with those who were detected by our studies, you can see nearly about 30% had diabetes. This is among adults above 20 years in the Colombo district. And you can see nearly about 40% had pre-diabetes. So if you take like a urbanized area like Colombo district, only about 30% is having normal blood glucose regulation. So that level is the situation is worse to that level. Obesity, we thought that we were not an obese population a few decades ago. We know obesity is one of the major risk factors for most of the NCDs and including cancer. So we embarked on looking at cutoffs for defining obesity in Sri Lankans, and we showed that the cutoff should be much lower than what is existingly used by agencies like the World Health Organization. You can see the real risk of increasing cardiovascular disease occurs at a BMI of around 20-21. and a waist circumference of around 77, which is very much thinner levels. These for females, not much different. And then also we showed that even being a low middle income country, about 35% was either overweight or obese. And this is much commoner in the urban areas where it comes to about 50%. And actually, compared with that, you can see these are figures again from Sri Lankan Uncommunicable Disease Study done in 2018. Whereas you can see the prevalence of either overweight and obesity together, particularly in female, you can see about 70%. This is much, much higher than what we saw about 15 years ago. And these are the risk factors for obesity, quite traditional, but as well as ethnicity, but we were not able to do studies on diet at that time. Again, we looked at the prevalence of hypertension and showed its association, particularly with the age. And you can see the data for hypertension again now, close to about 50% of the adult population is becoming hypertensive. And we look at the adequacy of control and other risk factors which are important, like smoking. Again, showing that it's predominantly a male problem. Ischemic heart disease, this is actually not the incidence, but the prevalence, which we'll underestimate because some people die before coming to hospitals. Again, however, you can see that with increasing age, it's also becoming a huge problem. So these are all to be anticipated to get worse because we are a rapidly aging population. Also, developing younger researchers, I was able to continue some more work on nutrition and also looking to see whether endogenously available things like cinnamon can be used to manage diabetes. There's some initial quite promising data on cinnamon, on diabetes and lipid control, which we have published. Then looking at interventions using mHealth, digital health, and also particularly looking at complications like neuropathy and its effects on falls. And in this paper published in the Journal of Epidemiology, we were able to describe the lipid issues, lipid patterns in Sri Lanka. So in addition to what we have already done, we wanted to see what new areas of research we need to undertake. We wanted to see whether we can prevent diabetes in the community. So we are part of a larger group of researchers from UK, Pakistan, India, and Sri Lanka together, looking at the prevention of diabetes in the community level with lifestyle interventions. And then also the studies have shown that if you lose a lot of weight, particularly at the initial stages after development of diabetes. Now here you can see, this is the amount of weight loss on the x-axis and the percentage remission means being free of diabetes. So if you lose about 15 kilos, there's a chance about 86% being free of diabetes. So we have embarked on work on these remission studies to see whether that can be a paradigm shift in the way we manage diabetes. Then came the COVID, and we wanted to look at things like how to manage and prevent COVID and COVID deaths in people with diabetes and the impact on the hormone system, the endocrine system. And also, when it comes to research, it is not a good thing just for you to do research alone. It is very important to nurture and mentor future generations so that you can really multiply by several fold the research output. So in that way, I had been able to get a lot of good, bright people trained in research, and most of these people are either specialists in medicine or eminent researchers in Sri Lanka or elsewhere in the world, also still having collaborations with us. And ladies and gentlemen, in conclusion of my particular subject today, we are having a huge epidemic of diabetes and other NCDs, which were underestimated by international agencies, but happy that we were able to correct with endogenous data. And we were able to contribute to the risk factors and also etiological subtypes and genetics and understanding of molecular mechanisms. And some of which I'm quite sure led to the subsequent developments in the Ministry of Health, where they established the NCD unit. Now it's a bureau working on the NCD, understanding the importance of NCDs, and then subsequently led to the revision of the WHO and IDF estimates and projections, particularly in diabetes, using our own data. And then a lot of modifications happened in the healthcare system, particularly aiming at the NCDs, like the healthy lifestyle centers and so on, and also stimulating more practical and cost-effective methods evaluation for early detection and prevention of diabetes and NCDs. Now, let me just briefly, before I wind up, tell a little bit of my involvement and my journey with IFS. So actually, when I was finishing my school education, I had a dream to be a doctor and a scientist, and one of the role models was none other than Professor Cyril Ponnamperuma, who was a world-renowned scientist who had brought a lot of fame to Sri Lanka. And then it was such a coincidence that there was closure of the schools for 2 years due to the problems we had in late 80s, where I had the opportunity to arrive at IFS. Here you can see the students at that year who got 4 A's for all subjects in all the disciplines with Professor Ponnamperuma and Dr. Arthur Clark, where we were brought in and given opportunities to work in research and has early research exposure. Not only research, actually, we were involved in organizing science parks in Gammudava. This is in the Kamburupitiya Gammudava site, where I was, manning IFS stall, where this was actually also made by ourselves. And then it's not only the work and research, we also had a lot of fun. You can see some of very prominent medical consultants and engineers in the country, researchers, and you can see the co-chair of this symposium, Professor Deepal here. Some of the others are scattered in the rest of the world. And again, a similar picture here. And so I'm quite grateful to IFS and other mentors like Professor Ponnamperuma, Professor Shanti Mendis, and from my own department like Professor Sharif, Professor Saroj, Professor Sham Fernando, and from the University of Oxford, and a few institutions, including where I started my school education, Royal College, then the IFS. From there, I went to the Colombo Medical School, the university, and then the National Hospital of Sri Lanka, the University of Oxford, all of which has helped me to achieve many things in research, as well as particularly other accolade like my Vidya Jyothi title. So ladies and gentlemen, we are in a situation like this where a tsunami of diabetes and NCDs is going to overwhelm us. Now, here actually people might not have much of a choice rather than getting to know early and fleeing the city to save lives. But in this tsunami of diabetes and NCDs, if we stand tall and strong and work together, we should be able to fight this NCD and diabetes tsunami. And that is where the role of the researchers also becomes important and very, very, should be appreciated. And we should all try to promote basic research, clinical research, applied and implementation research to find new answers to these ever-increasing issues in diabetes and NCDs. And I would like to again thank the chairman, the director, and the organizing committee, and all of you for inviting me, as well as the patient listening. Thank you very much.